Hello, my name is Leshem. I've done my med school internship in Tel Aviv University and Tel Aviv Medical Center. I'm currently pursuing a PhD in the Rani Linav's lab in the Weizmann Institute of Science. We study host microbiome interactions in various clinical setups. And after I finish my PhD, I intend to pursue a general surgery residency. And I'd like to thank the organizing committee for inviting me, and especially Professor Weizmann. This is an electron microscopy photo of intestinal villi with a huge amount of microbes surrounding it. When a baby is born, he's born without any microbiome, and he acquires his first microbiome from his mother when he goes through the birth canal or by contact with her skin if he is born through a C-section. We know today that the intestinal microbiome shapes our inflammatory or infectious response or our tolerantic response to various immunological stimuli. The microbiome diversifies throughout the first years of life until it reaches a stable, diverse composition. And when we reach adulthood or an older age, it starts to deteriorate approximately after the age of 70. There are multiple microbiomes, but today I'm going to focus mainly on the gastrointestinal microbiome, which is the most important microbiome and also the most relevant to the people who sit in this room. There are different microbiomes in different locations of the gastrointestinal tract, depending on the environmental conditions, on the pH, the oxygen content. As you know, most of the gut bacteria are anaerobic. And we know that, for example, in surgery, when we expose the gut lumen to oxygen, the microbiome signature changes and aerobic bacteria flourish. It's important for me to communicate the message that the microbiome affects distant organs. So certain environmental exposures change the microbiome, such as diet, systemic painkillers, surgery, and various other exposures. And the microbiome secretes metabolites. I'm not going to go into mechanisms too deeply, but they secrete metabolites that reach distant organs such as the brain and affect them, both in health and in disease. One of the hallmarks of the gut microbiome is huge, high inter-individual variability, which poses a real challenge in microbiome research, but also opens opportunity to personalized medicine, personalized patient care. Here you can see daily sampling of my stool microbiome in specific single individual throughout a whole year. And as you can see, throughout this year, the microbiome of this person was pretty much stable in terms of composition. This is except for a short period of time, around day 100, in which he traveled from a developed country to India. And as you can see, there's a change in the microbiome composition. Such a change we term dysbiosis. In this case, it was temporary, but sometimes it's permanent, depending on the nature and the severity of the perturbation. Another important message that I want to convey is that some known human pathogens reside within the gut in low abundance, and they do exist there, and they're usually dormant. However, under certain environmental conditions, exposures to broader spectrum antibiotics or surgical stress, they may bloom and cause infection. And this is the case of Clostridium difficile, which you are all very aware of. And this is a study from 2013 that compared fecal microbial transplant for the treatment of recurrent Clostridium difficile infection, and it showed very high cure rate of the FMT arm and compared to the standard care that was vancomycin. These high resolution rate of the infection were accompanied by an increase of the recipient's, the patient's bacterial diversity, suggesting that this bacterial diversity is the thing that's preventing from Clostridium difficile to infect us and to cause harm, and also that the diversity is what's important in FMT. Interestingly enough, this study was stopped prematurely because of high cure rates, basically because of high efficacy of the FMT. And patients who failed antibiotic treatment, which occurs often in Clostridium difficile, they were transferred to the FMT arm. This is a very interesting study that basically also used FMT, fecal microbial transplant, to treat Clostridium difficile. However, they took a slightly different approach where they filtered the fecal samples so as they didn't contain any bacteria. So basically, they treated patients with sterile fecal filtrate and reached cure of patients with Clostridium difficile infection. This was a proof of concept style, and the samples, this was a proof of concept trial, and the sample size was 5, but 5 out of 5 patients were cured, and this suggests that the non-bacterial component is what's mediating the beneficial effect, the therapeutic effect, and this may be a metabolite or a bacteriophage, which is a virus that infects bacterial cells. FMT is being evaluated in various indications. It's been shown it has shown promising results in ulcerative colitis in many studies. This one was just published recently. The situation in Crohn's disease is more complex, and the results are less promising. This is a list of ongoing clinical trials to evaluate the efficacy of FMT on various gastrointestinal and non-gastrointestinal conditions, and I've highlighted in green the surgical indications. And these are just indications that are being evaluated currently. One of the important tools and the interesting tools that we use when we study microbiome are germ-free mice. Germ-free mice are mice that are born and raised under a completely sterile environment inside isolators, and they have no indigenous microbiome, which allows us to perform interesting experiments with them, because if you take conventional mice and you transplant them with microbiome, there will be colonization resistance from their indigenous microbiome. However, when you transplant germ-free mice with a microbiome for humans or mice or any other mammal, the microbiome colonizes with relative efficiency, which allows us to compare. If we suspect a disease to be microbiome dependent, we can transfer a microbiome to germ-free mice and compare the microbiome of healthy and sick individuals by phenotyping those mice that received the FMT. One example of such study design was the transplantation of an obese microbiome. So basically, microbiome from obese mice was transplanted into germ-free mice, which became obese. They were lean and they became obese, whereas controls, the microbiome from lean mice, did not become obese. This was repeated many times with microbiome of both humans and mice in various setups, and also to show, basically to show that the microbiome mediates obesity. And we actually know today mechanistically that the microbiome is involved in many, many aspects of metabolism. Now, in this specific study, the mechanism that was suggested was that the microbiome of obese individuals had increased capacity to harvest energy from the diet. But I'm not going to go deep into this because we don't have time. I want to present a few studies that were done in the Rani Linav lab. Professor Rani Linav is an MD, PhD, and as an MD, he cares more about translational stuff and less about, basic science, structure of enzymes, etc. And I want to mention a few studies on prebiotics, which are substances that change the microbiome in a beneficial way that later can be beneficial to the host. And we mainly, when we say prebiotics, we mainly mean diet. And I want to also mention a few things about probiotics and fecal microbial transplants. The first study talks about current paradigm that diet should be defined by food. You're all pretty well aware of glycemic index. This concept means that a certain food has a certain glycemic index, and that some foods has low glycemic index, and therefore upon eating them, they will cause a low surge or low glycemic response in the blood glucose of the individual that consumes them. Whereas if a patient eats a food with a high glycemic index, he will have a high glycemic response. But data accumulates to suggest that this is actually more complex and more individual, person-specific, because patients who will eat the exact same thing will have completely different glycemic responses. And large body of evidence has supported this concept. And so in order to evaluate what would be the specific glycemic response of specific individual to specific foods, a study that was carried out in the lab I'm working by my colleagues, they recruited 800 participants, and they connected them to continuous glucose monitoring, which is a sensor that sits in the subcutaneous tissue and measures the interstitial glucose levels every few minutes. And they were connected for 1 week, in which they were monitored for their glucose levels, as well as the food they ate was put into a smartphone app Their microbiome was sequenced, and various blood and clinical other anthropometric parameters were also collected. During this week of follow-up, they ate real-life meals in addition to standardized meals, which we knew exactly what they contained and what amount of calories, carbohydrates, fats, etc. They contained in order to compare the response of different individuals to exactly the same meal. All in all, more than 130,000 hours of measurements were collected, and more than 1.5 million glucose measurements were also collected. And all this data was was used to train a machine learning predictor algorithm that its aim was to predict the glycemic responses of specific individual to a specific food. And before I show you the results and how accurate was the algorithm, this is how accurate is the prediction of the glycemic response of individuals based on their meal carbohydrate content, which is a state-of-the-art prediction before the study, because this is exactly the concept of glycemic index, as I showed. And after this 800 participant data was put into the algorithm, this predictor algorithm, the correlation between the predicted and the actual measured glycemic responses increased and almost doubled. This was also later validated by another cohort of an additional 800 participants. By the way, these were healthy participants. They weren't diabetic. These results were later validated in an American cohort of 330 non-diabetic American individuals. And this is important because some results that we find in specific areas of the world don't always recapitulate in other environments because the microbiome in different continents, in different lifestyles, and different cultures are somewhat different. As I say, the high individual diversity plays a role here and poses a challenge. And some of you may have heard of Day 2. This is a company that was founded based on these algorithm abilities to provide with patients diet recommendation based on their microbiome and other clinical parameters. And I forgot to mention that the original study, it was shown that participants who were prescribed with a diet with food suggestions based on their specific glycemic responses were capable of lowering their levels of glucose and basically improving their hemoglobin A1C. Another study that came out from the lab I work at by brilliant colleagues of mine wanted to evaluate probiotics. Probiotics are basically everywhere, and I'm not going to be surprised if people who sit in this room regularly or non-regularly prescribe probiotics to patients, although data to support the prescription of probiotics is very, very conflicting and very controversial. And so just before I present the study itself, this is actually it's two papers published back-to-back in Cell. I'm going to highlight the controversy by showing you what the reviewers of these papers said to us. Reviewer one said, "This is an unbalanced and poorly presented. What a pity the authors chose to go this route. This poor terminology, deceptive arguments. This is not the author's finest work. They are capable of better than this." And reviewer two said, "The most rigorous and well-written thesis on the field of probiotics I have ever read. The writing is excellent. No stone left unturned. I was turned into a student and glorified copy editor. Exceptional work." And so the study was designed to see what happens to probiotics inside the gastrointestinal tract throughout the entire length of it. And because most randomized controlled trials, and there are many, many randomized controlled trials of various empiric probiotics in various indications, and a lot of meta-analyses with conflicting results, most of them do not look what happens with probiotics. Are we colonized by them? Are we affected by them? Is the gut microbiome affected by them? And so this is the study design. Healthy individuals were randomized, were characterized at baseline, and then were randomized to receive either a course of probiotics for 28 days or placebo, which was obviously identical to the probiotics. And they were invasively characterized by colonoscopy and gastroscopy at the beginning, at baseline, and during probiotics treatment. And mucosal and luminal samples were collected, and we know that the mucosal and luminal microbiome are a bit slightly different. And biopsies were taken for RNA sequencing to evaluate the host transcriptome. These are the anatomical locations from which samples were taken, and as you can see, this is a very dense sampling strategy, including duodenum and terminal ileum. And I can tell you from now that regarding the question, are we colonized by probiotic strains? The patients that were treated with probiotics were roughly divided into two groups. One was the permissive group, which were partially or completely colonized by probiotic strains, and the other group of patients that were resistant. And resistance or permissiveness could be predicted by their baseline microbiome signature. Another important finding was that, as I told you, and this was already known, the gut microbiome composition in many various different gastrointestinal locations is different. But here you can see that the stool microbiome on the right is different even in the most distal locations of the gastrointestinal tract. As you can see, the descending colon, the rectum, and the sigmoid colon. By the way, in the group of the resistant participant to probiotics, probiotics were shed in the stool during the probiotic consumption, but later when they stopped taking probiotics, the probiotic strains were not in the stools. And many studies that evaluated probiotics concluded that that the probiotics were colonized if they saw probiotic strain in the stool, but this basically means that they're being shed, not necessarily that they colonized the GI tract. And then they said, the same group of investigators, what happens if we clear the mucosal niches from the endogenous bacteria, which conveys resistance, by antibiotic exposure, broad-spectrum antibiotics, and then give them probiotics. And so to that, and they hypothesized that this may facilitate colonization. And so to that aim, again, another group of participants were evaluated at baseline, were then exposed to 1 week of antibiotics, cipoflagil, and then they were randomized to receive either probiotics or autologous auto-FMT, which means basically that the person receives his own stool from before antibiotic exposure, or nothing, basically the watchful waiting group or the spontaneous recovery, if you will. And again, patients were characterized invasively by gastroscopy and colonoscopy to see what happens throughout the gastrointestinal tract. And there's a lot of data in this study, but I'm only going to show a small piece of it. In this graph, you can see the number of observed species throughout time. And as you can see in red, this is the spontaneous recovery group. And basically in all group, antibiotic exposure decreases the microbiome diversity. And then in the spontaneous recovery group, the microbiome starts to recover slowly. But in contrast, probiotics, not only that they don't improve and enhance the reconstitution of the baseline microbiome, they actually delay it. And this is interesting because one of the most common scenarios in which probiotics are currently prescribed is in clinical settings is after antibiotic exposure. And as you can see, not just that probiotics don't benefit the patient, they also harm. And in sharp contrast, the autologous FMT was excellent at reconstituting the baseline microbiome of patients very rapidly. And I'm not going to show it, but actually the host transcriptome, the RNA expression profile, also normalized quickly in the auto-FMT group, better than the spontaneous recovery or the probiotics group. And now I want to share some data on microbiome in surgery. There's not a lot of data, and I'm not going to show all of it, but I'm going to show parts of it. Basically, the perioperative management is not good for the microbiome. We expose patients to antibiotics, mechanical brow preparation, anesthesia, systemic painkillers. These all decrease the diversity of the microbiome, which I've already showed you is important. Early recovery after surgery protocols, ERAs, they're good from the microbiome, but this I only say out of common sense. This was not evaluated empirically, but it makes sense that if we decrease the amount of the time the patients are under nothing per os or the amount of opioids they receive, their microbiome is going to be less perturbed. Yeah, I'm going to go, I wanted to talk a bit about the STOPI trial. I'm going to skip that and go through straight to microbiome in postoperative complications. This is a rat model in which certain bacteria were found to be involved in nitric oxide production. Nitric oxide paralyzes smooth muscle and induces ileus. And this could be abrogated by targeting these specific bacteria. And again, this is a rat model. Another study found that also started from a rat model and showed that rats that had anastomotic leakage contained 500-fold increase in Enterococcus fecalis, and they later found also that Enterococcus fecalis has the capacity to induce host enzyme that degrades collagen, and that the amount of Enterococcus fecalis correlates with the collagen degradation capacity. They later go on to show that humans who had anastomotic leakage also had high amounts of Enterococcus fecalis and also high collagen degradation capacity. Another study published in the Annals of Surgery claimed to have found a predictive microbiome signature that can predict the risk of colorectal anastomotic leakage. However, this study had several methodological flaws, and I'm not going to go into that, just to give you a taste of what's being looked at in this field. A little bit about the microbiome in cancer. The microbiome is known to be involved more or less in every aspect of cancer. Patients with colorectal cancer has altered microbiome. They have dysbiosis. And microbiome sequencing is being evaluated as a screening method for colorectal cancer, but recently 2 studies were published, but not with great success, and so I'm not going to present them. And also I think that using sequencing as a screening method is kind of futile because it's really expensive, and we need the screening test to be not so expensive. This is a survival curve of patients with various cancers, in this case non-small cell lung cancer, renal cell carcinoma, and urothelial carcinoma, and you can see that antibiotic exposure was associated with poor prognosis. Now, I know what you're all thinking. You're thinking that, all right, these patients had infections, and so they died because of their infections, not because of antibiotics that changed their microbiome, and I agree. It's what I was thinking when I read this paper. However, some papers show that mechanistically the microbiome is very much involved in response to cancer treatment in oncology. And I'm going to show only one example, although there are many. This study utilized this model of performing fecal microbial transplant into germ-free mice. And it compared transplant of microbiome from patients who responded to therapy to patients who did not respond to therapy. And as you can see in the 2 blue plots, when germ-free mice were colonized with microbiome of non-responder patients and treated for their cancer, in this case this was melanoma, there was no response to therapy. But in contrast, when germ-free mice were colonized with responders microbiome, there was response to therapy. And this was, again, demonstrated in various cancer models in mice, but I'm not aware of any clinical trials that evaluated FMT in humans as a way of inducing patients' response to therapy. And by the way, in this study, the treatment was PD-1 blocker, which is an immunotherapy. Now to a cancer that's a bit more relevant to you, pancreatic adenocarcinoma. A study that came out by the Ravid Straussman lab in the Weizmann Institute of Science showed that there are bacteria that reside within pancreatic tumors that have the capacity to degrade gemcitabine and confer basically resistance to therapy in patients that are treated with gemcitabine. Specific bacterial strains were identified, mainly Mycoplasma, but not just Mycoplasma. And they later go on, they show this in a mouse model, but then they go on and show that in human tumors, these bacteria exist as well. And here in this figure, you can see the bacteria in red among cancer cells in blue. And interestingly, these bacteria were more abundant in patients that their pancreas were instrumentized. I want to finish by discussing bariatric surgeries. This is what I'm doing in the lab, and you're all aware of the benefits and problems with bariatric surgeries. You should know that bariatric surgeries change the microbiome depending on the type of procedure, but pretty much all procedures change the microbiome very rapidly, starting a few days after surgery, and this change exists in the long term. This is human data on change in microbiome composition before and after therapy. This is data from a mouse model of Roux-en-Y gastric bypass compared to sham and weight-matched sham, and as you can see, the microbiome composition changes. But I'm focusing in my PhD more in a narrower question. I'm specifically focusing on early diabetic remission after bariatric surgery, and I want to understand what's the mechanism behind the fact that patient glucose levels improve significantly right after surgery, even if they stop any medication. It seems that their insulin sensitivity improves. And there are many mechanisms suggested, but no one knows the exact mechanism. And here you can see data from a clinical study that compared patients who underwent bariatric surgery, either gastric bypass or sleeve gastrectomy. And you can see that a glucose tolerance test, amasat sukar, was improved within 3 days after surgery in the two left panels, which did not occur in the right panel, which was a group of patients that underwent caloric restriction in a manner similar to surgical patients. This is a study that showed that if you take a mouse model of vertical sleeve gastrectomy, if the mice are treated with certain antibiotics, it was shown with 3 different antibiotics directed at gram-positive bacteria, the beneficial effect of surgery in the short term are abrogated and ameliorated by antibiotic exposure, which suggests that the microbiome is involved in some of the metabolic improvements in the early postoperative period after bariatric surgery. And there are many more evidence, which I'm not going to show because I don't have the time. And so I've developed a vertical sleeve gastrectomy model in type 2 diabetic mice. I'm not the first one to do it. Several models exist, but I've taken them and modified them with the help of Dr. Nir Choresh, who sits here. But basically, we take a mouse, we open it, we take the stomach out, and then we apply a hemostatic clip on the stomach and exclude the part that that should be excluded in sleeve. And this is a mouse. Don't know if it's going to work. Basically, there's. אתם מפעילים את זה או אני? בסדר. This is a mouse less than 1 day after surgery. It looks quite well. If we're able to decrease the amount the mouse are on fasting and then basically decrease the amount of stress and go into the abdomen by a very small cut than what is just suggested in most protocols. And in my model and in other models in the literature, the metabolic improvements in mice are very similar to humans in terms of weight, glucose sensitivity, insulin sensitivity, and other things. You can see here that in red, in baseline, I have diabetic mice who have high glucose surge in glucose tolerance test compared to healthy controls in blue. And you can see the area under the curve in the column plots above. And as you can see, as early as 1 week and even earlier, and I'm not showing this here, the glycemic response of these mice improved significantly. And it seems that their insulin sensitivity significantly improved. And the control mice underwent sham surgery. We are now also working on designing a human cohort. I'm doing this with the help of David Goiting and Dr. David Goiting and Dr. Nir Choresh. We're going to recruit diabetic patients who are going to undergo bariatric surgery and connect them to continuous glucose monitoring at baseline and after surgery, and characterize their microbiome throughout a period of time in the early postoperative period. And also we're going to use metabolomics and RNA sequencing to characterize what's going on and try to decipher the mechanism behind early diabetic remission. So the take-home message is that the gut microbiome, to my opinion at least, has some significance in health and disease. Data on the involvement of microbiome in surgery is lacking, and I think this is a great opportunity to explore unexplored areas. And I think that basic science may help where randomized controlled trials haven't so far, where years of debates regarding preparations of patients to surgery, yes, no, mechanical brow preparation, the type and the length of antibiotic exposure. I think that we can use basic science to understand mechanisms and have better answers and provide patients with tailored, personalized perioperative care instead of giving them care bundles like ERAS or surviving sepsis, which are good but not personalized, and we don't really understand what we understand what we're doing, but I think we have the ability to go down into the details and understand some mechanisms and provide patients with better care with the help of basic science. And you don't have to work in a lab or have training of basic science. You can just collaborate. And I want to thank Professor Rani Linav, my mentor, Dr. Nir Choresh and Dr. David Goiting. I want to really thank General Surgery in the Tel Aviv Medical Center for welcoming me to practice surgery throughout my PhD during the weekends, and most importantly to all my lab members. Thank you.